In part 1, I had dealt with the central dogma and transcription in prokaryotes. In part 2, I had explained about the transcription in eukaryotes. In part 3, I had explained about the translation in prokaryotes. And in this part 4, I'll be explaining to you the translation process in the eukaryote. So this is Professor R.M. Mahendrakar from JSS, Arts, Science and Commerce College, Gokak. So let's begin with this topic, translation in eukaryote. Translation is a process in which the protein is synthesized from the information contained in the molecule of messenger RNA. It is defined as the process by which the sequence of nucleotides in mRNA molecule directs the incorporation of amino acids into proteins. The machinery required for translating the language of mRNA into the language of protein is composed of following primary components. First one is mRNA, which carries the genetic information of DNA in form of codons. Second is transfer RNA. It acts as an adapter between the codons and the amino acid they specify. Third, amino acids, which are present in the amino acid pool of the cytosol. And the enzyme, that is amino acyl tRNA synthetase and peptidyl transferase, and the factors. Now, in eukaryotes, we have 13 factors, of which 10 are the eukaryotic initiation factors, 2 are eukaryote elongation factors, and 1 eukaryote releasing factor. Unlike in prokaryotes, where we had 9 factors. 3 for initiation, 3 for elongation, and 3 for the releasing. Whereas here in eukaryotes, we have 10 for the initiation process, 2 for elongation process, and 1 for the releasing process. So first let us start with the first step, that is the initiation process. The initiation of translation in eukaryotes is complex, involving at least 10 eukaryotic initiation factors. And this whole process is divided into 4 steps, namely ribosomal dissociation, second formation of 43S pre-initiation complex, third formation of 48S initiation complex, and fourth formation of 80S initiation complex. So first let us see the ribosomal dissociation. The 80S ribosome dissociates to form 40S and 60S subunit. The two initiation factors, namely the EIF3 and EIF1A, binds to the newly formed 40S subunit and thereby block the reassociation of the 60 subunit. Now here the E stands for eukaryotic, I stands for initiation, F stands for factors. The next process of initiation is formation of 43S pre-initiation complex. Ternary complex containing of the methionine transfer RNA and initiation factor 2 bound to GTP attaches to the 40S ribosomal subunit to form a 43S pre-initiation complex. Now here you can see, now this is the initiation factor 2 which binds with the GTP molecule, then this complex binds with the transfer RNA carrying the methionine amino acid, because in eukaryotes the first amino acid that is attached is the methionine, unlike in prokaryotes where it was N-formyl methionine, whereas in eukaryotes it is only methionine amino acid. So that's why we call this as met-tRNA. So now we get this complex which consists of the transfer RNA carrying the methionine amino acid, initiation factor 2 and the GTP. Now this complex we call it as ternary complex. Now this ternary complex now binds with the 40S subunit and results in formation of a 43S pre-initiation complex. So this is the second step. The third step is formation of 48S pre-initiation complex. I hope you know what is the meaning of S. S stands for the Swedberg's unit. It is a sedimentary coefficient. The binding of mRNA to the 43S pre-initiation complex results in formation of the 48S initiation complex through the intermediate 43S initiation complex. Here, initiation factor 4F complex is formed by the association of initiation factor 4G, initiation factor 4A, and initiation factor 4E. So all these result in formation of this initiation factor 4F. Now this initiation factor 4F, which is referred to as the cap binding protein, because this binds with cap at the 5' end, which is the common feature we see in the eukaryotic mRNA. So because in eukaryotes, already I have told you, during the post transcriptional changes, the 5' end is added with a cap, then the whatever the introns are there, they are removed, only the exons are kept, so splicing occurs, and at the 3' end there is a long adenylate tail. So these are the three important post transcriptional changes that occur in the mRNA of this eukaryote. So now this initiation factor 4 binds with the cap of this mRNA. Now after the binding of this cap binding protein to the mRNA, this mRNA is then transferred to the 43S complex. After the binding of the initiation factor 4F to the cap end of the mRNA, the initiation factor 4A and initiation factor 4B bind to mRNA and reduce its complex structure. This mRNA is then transferred to 43S complex. For the appropriate association of 43 pre-initiation complex with mRNA, energy has to be supplied by ATP. The ribosomal initiation complex scans the mRNA for the identification of appropriate initiation codon, that is AUG. So this process we call it as mRNA, and in the meantime the larger subunit, that is the 60S subunit, binds with the GTP molecule with a factor 5B. Now this results in formation of a 80S initiation complex. The 48S initiation complex binds with the 60S ribosomal subunit to form a 80S initiation complex. The binding involves the hydrolysis of GTP to GDP. This step is facilitated by the involvement of this initiation factor F. As the ATS complex is formed, the initiation factors bond to the 48S initiation complex are released and recycled. Again, we shall just recall the initiation process in eukaryotes. First, initiation factor 2 binds with GTP, and this complex then binds with the transfer RNA carrying methionine amino acid and forms this complex. This complex now binds with the smaller subunit of the ribosome, that is 40S. 40S is linked with this initiation factors 1, 1A and 3, because this binding is essential to prevent the 60S subunit from joining it. So then after this complex is formed, it binds with the ribosomes and results in formation of 43S pre-initiation complex. Then the complex 4F, which consists of the 4E, 4G, 4A, 4B, all these factors result in formation of this initiation factor 4, which binds with the cap end of the mRNA, and this after the attachment of the factor 4 with the cap end, now the mRNA binds with the 43S pre-initiation complex and forms 48S initiation complex. Now, once this 48S initiation complex is formed, then the 60S subunit binds with the smaller subunit and releases all these factors. These factors which are released, again they are cycled back, and then this, after the binding of this 60S subunit, now this becomes a 80S initiation factor, and thus the process of initiation ends here. Now we go to the next, that is elongation process. Now whatever the energy that is needed for the initiation process, it is provided with these ADP molecules and GDP. So next we shall see the elongation process. Now this elongation process is almost same as what we had seen in prokaryotes. So here the first the transfer RNA is occupied at the P site. Okay, the ribosome elongates the polypeptide chain by a sequential addition of amino acids. The amino acid sequence is determined by the order of the codons in specific mRNA. Elongation, it is a cyclic process which involves the three major events. That is, first one is amino acyl tRNA binding at A site, then peptide bond formation between the newly arrived amino acid and the pre-existing amino acid, and then the dislocation of the transfer RNA after the supplying of the amino acid. So here you see a new codon has appeared at the A site. A transfer RNA with a complementary pairing comes and attaches here. Okay, and then once the transfer RNA with the corresponding complementary pairing comes and attaches here, then the peptide bond is formed between the pre-existing amino acid and the newly arrived amino acid, and this is facilitated by this peptidyl transferase enzyme. Then after the formation of the peptide bond, then the ribosome moves again one codon length from 5' to 3' direction. So now at the A site there is a new codon, and the first arrived transfer RNA is at E site. So once it comes at the E site, the transfer RNA is released and the cycle continues. So like this, in this elongation cycle, these three events occur. One is the amino acyl tRNA binding at A site, peptide bond formation between the pre-existing amino acid and the newly arrived amino acid, and the translocation of the transfer RNA at the E site. So this cycle continues and the elongation continues till the A site gets occupied with the stop codon. So once the A site of the ribosome gets occupied with any one of these terminating codons, either UAA, UGA or UAG, the termination process begins. Now, in case of the eukaryotes, there are only two elongation factors and only one releasing factor. So one of the stop codon or termination signal terminates the growing polypeptide. When the ribosome encounters a stop codon, there is no transfer RNA available to bind with the A site of the ribosome. Instead, a releasing factor with a GTP molecule comes and binds at the A site of the ribosome. In eukaryotes, there is a single release factor. The eukaryotic releasing factor, that is ERF1, recognizes all the three stop codons, and the releasing factor 3 stimulates the termination event. Once the release factor binds, the ribosome units fall apart, releasing polypeptide chain, the transfer RNAs and the mRNA, thus brings end to the process of this translation. To participate in the new round of polypeptide synthesis, these mRNAs and tRNAs must be released, and the ribosomes must dissociate into small subunit and large subunit. Collectively, these events are termed as ribosome cycling. Now let us compare between the translation process of prokaryotes and translation process of eukaryotes. We see that the overall mechanism of protein synthesis in eukaryotes is basically the same as in prokaryotes, except some significant differences. And these are: a prokaryotic ribosome has a sedimentary coefficient of 70s and the subunits of 30s and 50s subunit. Whereas a eukaryotic ribosome has a sedimentary coefficient of 80s and the two subunits, the larger subunit is having a 60s and the smaller subunit is having 40s sedimentary coefficient. Composition of eukaryotic ribosome subunit is also more complex than prokaryotic subunits, but the function of each subunit is essentially the same as in prokaryotes. The next main difference between the initiation of translation in prokaryotes and eukaryotes is that in bacteria, a Shine-Dalgarno sequence lies at the 5' end to the AUG initiation codon and is the binding site for the 30th ribosomal subunit. In contrast, most prokaryotic mRNA do not contain Shine-Dalgarno sequence. Instead, a 40th ribosomal subunit attaches at the 5' end of the mRNA and moves downstream until it finds the initiation codon. This process is called as scanning. In eukaryotes, each mRNA is monocistronic, that is, the mRNA contains a single gene, whereas in prokaryotes, the mRNA is polycistronic. It contains more than one gene, that is, the mRNA encode several proteins. Each coding sequence in a prokaryotic mRNA has its own initiation and termination sequence. The initiation of protein synthesis in eukaryotes require at least 9 distinct eukaryotic initiation factors compared with only 3 initiation factors in prokaryotes. The next main difference is in eukaryotes, the initiation codon is methionine, whereas in prokaryotes, the initiation codon is a special amino acid, that is N-formylmethionine, and this is brought about by a transfer RNA called as transfer RNA F-met. Prokaryotic translation doesn't require helicase because there is no post-transcriptional changes in the prokaryotes, whereas in eukaryotes, post-transcriptional changes takes. That is, the mRNA that is formed undergoes processing and then it gives rise to a functional mRNA. The next basic difference is in prokaryotes, transcription and translation, they occur in the cytosol itself, whereas in eukaryotes, you see, the transcription process takes place in the nucleus, whereas the translation process takes place in the cytosol. So transcription is taking place inside the nucleus, translation is taking place outside the nucleus in case of eukaryote. The next is in prokaryotes, transcription and translation may become coupled, that is simultaneously both the transcription and translation can take place, whereas in eukaryotes there is no coupling. First there is a transcription process is followed by the post-transcriptional changes, then after when it comes into the cytoplasm, then only the translation process begins. So these are some of the basic differences we see in the protein mechanism in both prokaryotes and eukaryotes. So with this, we conclude this topic on protein synthesis both in prokaryotes and eukaryotes. Thank you. Please take care. Stay home. Stay safe. Once again, thank you.